Hello everyone, my name is Jonas Cohen. I work at the Station Biologique de Roscoff in France. I have worked most of my life with seaweeds, and I will talk about what I've been doing with algae containing carrageenans. So I will talk about how I worked with the carrageenophytes and a little bit use some insights from Chondrus crispus in how we can understand better the eucheumatoids. So my interest in eucheuma species started actually with my thesis. My thesis project was to be carrageenan biology in eucheuma species. So the idea was to go to Zanzibar and collect eucheuma and or Cavaphycus. Take them to the laboratory in Sweden and grow them and study effects of growth conditions on the content and quality of carrageenans. This actually did not work. They all died. Apparently they didn't want to go from a tropical paradise to the frozen tundra in Sweden. So this actually meant that my thesis was on stress biology in seaweeds. And one of the things I started with was stress biology in eucheuma. So one of the reasons they died is, I think, that in their defense mechanism, there's an induction of reactive oxygen species. And this is caused by stress and could be different kinds of stress. And one example of a stress is mechanical stress. So this experiment is with a species called Eucheuma platycladum. So when you break this algae apart, it will actually produce reactive oxygen, in this case, hydrogen peroxide. This is caused by an enzyme that is called an NADP oxidase that will produce the hydrogen peroxide. Looking back, I think one of the major reasons that I didn't manage to grow with eucheuma in Sweden was that I didn't add any antibiotics. People that have successfully grown eucheuma in the laboratory have typically used antibiotics. So I think that's the major reason they did die. After a period not working so much with algae using carrageenan in their cell walls, almost 10 years later, I started working with a new model, which was Chondrus crispus, a little bit with similar questions about stress, but trying to use more modern methods, looking more at the molecular biology of the species. So one of the first things I did when I arrived in Roscoff was to look at the NADP oxidase in Chondrus crispus. So in Chondrus crispus, you can have an oxidative burst in the interaction between Chondrus crispus and the green alga. And part of the gene was obtained through PCR. And because this is 20 years ago, we actually used this PCR product to screen a cDNA library. It took me something like 3 months, but eventually we got the gene. So this was not very efficient. So how should we do to advance faster? And our answer was the Chondrus crispus genome project. So we decided to sequence Chondrus crispus. So we sequenced a haploid gametophyte. We found a genome of around 105 megabase pairs, a little bit more than 9,600 genes. And at the time, over half of these genes were unknown from other organisms. Now with much more red alga sequence, we have a little bit more genes that are also found in other organisms. Just as an example, with my screening project, I found one NADPH oxidase. Looking at the genome, we actually know that there are 4 of them. Some of the insights from the Chondrus genome was that genes are clustered. So genes are sitting in regions where they are physically close on the genome. And then you have areas in the genome with has a very low gene density. The genes are compact, meaning that there are very few introns. There is 9,600 genes, are not that very many for multicellular eukaryote, so there is a limited number of genes. It seemed to be a normal but minimalistic eukaryote. And one of the things that are important is that they are typically small gene families. So it's very often that you have only one gene for one enzyme. So back to Carrageenan. Can we use genomic and transcriptomic data to understand carrageenan metabolism? The first experiment I want to show you is an experiment that was based on the idea that we should take Chondrus crispus and we should make protoplast. So protoplast are cells where we have taken away the cell wall. So we degraded the cell wall with enzyme capable of degrading carrageenan, cellulose, and the other compounds, and produce protoplast. So the idea here was that the protoplast would have a strong urge to produce a cell wall. So the idea was that with this overexpression of cell wall genes, it would be easier to find the genes important to make carrageenan. In reality, it doesn't work because protoplasts are extremely stressed plants. So what we saw instead was that we got much more genes that were important in stress. And we couldn't really find any genes that were specifically overexpressed in the protoplast. And one of the reasons for this is that they were actually not viable. So the protoplast slowly died. So I think what we were looking at was extremely stressed cells. This was a good thing because we could, by using this approach, look at stress responses in Chondrus, but it didn't give any insight into carrageenan metabolism. So we wanted to use another approach, and this approach is based on the fact that in the life cycle of Chondrus crispus, you have gametophytes and tetrasporophytes. So you can have female gametophytes and male gametophytes. If you go out on the shore during low tide, you can easily find tetrasporophytes that are diploid and gametophytes that are haploid. So this is the life cycle of Chondrus crispus. It starts with the tetrasporophyte that is diploid, that will through meiosis produce tetraspores that will grow into male gametophytes and female gametophytes or female gametophytes. That will the male gametophyte will produce male gametes that will fuse with the female gametes and produce carposporophytes that will produce carposporophytes that will produce tetrasporophyte. What is interesting here is that there's a difference in carrageenans in the tetrasporophyte and the gametophytes. So the tetrasporophyte contains mainly lambda carrageenan and the gametophyte mainly iota and kappa carrageenan. So the approach here was that we wanted to study which genes are important for the synthesis of carrageenan. So here you can see the synthesis of carrageenan. There are a number of galactosyl transferases, sulfotransferases that are needed, and also sulfurases. I said before that gene families in red algae are typically small. This is not totally true for the genes that are important for cell wall synthesis. So for instance, we have 17 sulfurases in Chondrus, but you have different sulfurases that will work on different parts of the metabolism of carrageenan. So the idea here was a little bit to try to see which genes are important for a specific reaction. And the idea here is that if we look at the sulfurases, this sulfurase here that makes kappa carrageenan would be much more expressed in the gametophyte compared to the tetrasporophyte. And in this way, we could potentially identify the important genes for the different types of carrageenans. So this is the result of 40 genes important. Unfortunately, what was to a large extent seen was that the tetrasporophyte, the much higher expression of genes important for carrageenan production. But we could see here for this gene, which is a sulfurylase, that is actually having higher expression in the gametophyte than the tetrasporophyte. So this means that this gene is potentially this gene because we expect this one to be higher expressed in the gametophytes, male and females, because they contain much more kappa carrageenan than the tetrasporophyte. So this gives you targets so you can continue doing biochemistry. And I think this is important, that we still really need to do biochemistry to prove what the different genes are doing and to understand carrageenan biosynthesis. And there is a lot of things we don't know. And one of the ways we could also do this is by comparing the genomes of different eucheumas. So different eucheumas or kappa ficus with different carrageenan would potentially have different genes or different combination of genes, and this could also help us to understand carrageenan biology. I would like to end by saying thank you for listening. I would also acknowledge that Elizabeth Ficoblin and Agnieszka Lipinska did a lot of the annotation of the carrageenan genes and a lot of the work with the different life cycle stages. Natalie Kovelchis was very helpful during her thesis for the stress experiments. The sequences of the genome was done by the Chondrus consortium, which was a lot of people, and funded by Genoscope, and many others have helped with the production of these data. Thank you very much. Thank you very much, and I hope you enjoyed it. Bye-bye.